Hello students, I welcome you to EPG Pathshala. My name is Dr. Shiv Kumar Sood. I am Senior Scientist in the Division of Animal Biochemistry at National Dairy Research Institute located in Karnal, Haryana, India. Today, I am going to talk about the module Sequence Alignment, a practical example from the paper Biostatistics and Bioinformatics. Dear students, there are several sequence alignment programs for global and local alignment of a pair of sequences. Embossed needle and embossed stretcher are freely available online for global alignments. Local alignment tools create an alignment covering subsequences of full-length subsequences. On the other hand, S-Search and BLAST, also freely available online, are creating local alignments. Users need to choose the most appropriate program for a given task in hand. In addition, users need to decide the scoring matrix as well as gap penalty scheme with input values for penalizing introduction of gaps. Then user need to draw an inference of homology from the sequence alignment. All this will be undertaken using embossed needle and embossed stretcher, the two programs for global sequence alignment. First, with a hypothetical pair of protein sequences, and then with a real practical example of HPR proteins from E. coli and Enterococcus faecalis. This will be followed by an example using S-Search and BLAST programs for local sequence alignment. Finally, users will undertake graphical and statistical evaluation of the alignment to draw an inference of homology. In the present module, the students will learn the use of global and local sequence alignment programs for a pair of sequences. Then a practical example of sequence alignment will be undertaken using these programs to draw an inference of homology. Embossed needle, available at European Bioinformatics Institute, creates an optimal global alignment of two sequences using the Needleman-Wunsch algorithm with affine gap penalty. Linear gap penalty is not implemented because linear gap scoring scheme is useful for understanding the basic dynamic programming manually as well as for using local alignments of DNA sequences, whereas the actual alignment of evolutionary related sequences is better achieved using affine gap penalty. There are separate interfaces for aligning protein and nucleotide sequences. Both differ only in the scoring scheme to be selected. There are 3 steps in alignment using embossed needle. Step 1 is to enter the input sequence for alignment in the provided text boxes. Users may paste the sequences or, using the browse buttons, upload saved files in the FASTA sequence format. In step 2, set the parameters for scoring. It offers to select BLOSSOM matrix from BLOSSOM 30 to BLOSSOM 90 with any increment of 5. The default is BLOSSOM 62. In addition, or on the other hand, one can select from M10 to M500 with any increment of 10. The allowed gap opening penalty values ranges from 1 to 100 and gap extend penalties ranges from 0.005 to 10. In the present module, the students will learn the use of global and local sequence alignment programs for a pair of sequences. Then a practical example of sequence alignment will be undertaken using these programs to draw and inference of homology. Embossed structure, also available at European Bioinformatics Institute, uses a modification of the Needleman-Wunsch algorithm that allows larger sequences to be aligned globally. With embossed structure, there are again 3 steps. Step 1 is to enter the input sequences in a similar interface as used for entering input sequences during embossed needle usage. Step 2 is to set the parameters for scoring. It offers to select blossom matrices from blossom 30 to blossom 90 with any increment of 5. The default is blossom 62. In addition, again, one can select from PAM10 to PAM500 with any increment of 10. However, embossed structure allows only a small range of opening, that is from 1 to 25, and extend gap from 1 to 8 penalties. Choose higher blossom number matrix with high gap opening and high gap extent penalties for aligning closely related sequences. For aligning distantly related sequences, choose lower blossom number matrix with low gap penalty and low gap open and extend penalties. When the divergence distance is not known or the sequences are having intermediate divergence, choose blossom 62 with 10 for gap opening and 1 for gap extend penalties. Alternatively, try all the 3 options to arrive at 3 alignments to select the best one. Now submit for sequence alignment. Using Smith-Waterman algorithm of dynamic programming, S-Search, available at Protein Information Resource, that is PIR, constructs a local alignment between full lengths of 2 sequences. It requires pasting 2 sequences only and no other input is required. In the present example, sequence 1 is an actual protein of 412 residues and sequence 2 again an actual protein with 411 residues. That shows, and they are a close homologous. But 159 residues were deleted at several points in sequence 2, resulting in a sequence having only 252 residues left in sequence 2. This deletion or a number of deletions at several points was introduced manually to simulate evolution over a longer divergence distance. This alignment shows that S-Search gives local alignments using Smith-Waterman algorithm of dynamic programming between full lengths of 2 sequences of even unequal lengths as envisaged during evolution. All the deletions at various points in sequence 2 has been aligned with residues in sequence 1 by introducing gaps. BLAST variants available at NCBI can also align 2 sequences using local alignment. Simply check the checkbox to use BLAST as an alignment program for sequences rather than similarity search program. Paste 2 sequences for pairwise alignment. However, keep in mind to match the BLAST variant with the type of sequence 1 with the sequence 2. For example, when using BLAST-X, sequence 1 has to be DNA and sequence 2 has to be protein. Click BLAST to receive results in the following window. Embossed Needle reported this alignment with Blossom 62 using gap opening penalty and gap extension penalty of 1 each. This alignment shows 14 identities with 1 conservative mutation, 3 semi-conservative mutations, and 5 gaps, having an overlapping alignment length of 23 with a score of 67. The identity between two sequences is quantitative and therefore can be used to infer homology from the percentage of identical residues aligned over the length of pairwise sequence alignment. In the present case, 60.9% identity over 23 aligned residues can be used to infer homology between these two sequences by using an XY graph. To infer homology between two sequences by using the graph, plotting the number of residues aligned versus percent sequence identity over the y-axis for proteins known to be homologous. All homologous proteins graph fall in green region. Therefore, if XY graph between the number of residues aligned taken on x-axis versus percent sequence identity on y-axis for two aligned proteins fall in green region, then it is safe to infer that these two proteins are homologous. This zone has been named as safe zone to infer homology. On the other hand, when the sequences are not homologous, then the XY graph will fall in the red zone. Therefore, if an XY graph between the number of residues aligned and percent sequence identity for the aligned proteins fall in red zone, then it is not safe to infer homology between these two sequences. This zone has been named as midnight zone. In the yellow zone, user need to be careful before inferring homology and must follow statistical assessment of sequence alignment. In the present case, with 23 residues aligned with 60.9% identities, the alignment falls in yellow colored twilight zone. Therefore, user must undertake statistical evaluation of this sequence alignment. The statistical significance of an alignment score is frequently assessed by calculating p-value to distinguish whether the alignment is due to homology or just by chance. The p-value is given by k e raised to the power lambda s, where k is natural scale for the alignment space size, that is total lengths of two sequences. E is the exponentiation function, lambda is a natural scale for scoring metrics used for alignment, and s is the raw alignment score. However, sequence alignment programs report only score for the alignment. Parameters k and lambda are not reported by these alignment programs. PRSS program, available online at M10, calculates the parameters k and lambda. Simply input scoring parameters and paste two sequences as plain text in the provided text boxes to run PSS. On the following result page, PRSS reported lambda equal to 0.2051 and k equal to 0.8993. Now p-value can be calculated using Microsoft Excel. PRSS program reported k and lambda values. Enter k, lambda, score and p-values as labels in the cells A to D in row 1 of MS Excel worksheet as shown in the slide. In row 2, enter actual values for these parameters, that is 0.8993, 0.2051 and 67 in cells A2, B2 and C2 respectively. In cell D2, enter the shown formula and press enter. The resulting p-value is displayed immediately in cell D2, which in the present case is approximately 10 raised to the power minus 6. This p-value shows that there is one chance out of approximately 1 million for this alignment to be just by chance. To infer homology from p-values, 5 ranges has been defined. When the p-value is less than 10 raised to the power minus 100, then the two sequences involved in alignment are identical sequences. When the p-value is between 10 raised to the power minus 100 and 10 raised to the power minus 50, then two sequences are nearly identical. When p-value is between minus 50 to 10 raised to the power minus 5, then two sequences are having clear homology. When p-value falls between 10 raised to the power minus 5 to 10 raised to the power minus 1 range, then two sequences are having possible distant homology. And finally, when p-value is more than 10 raised to the power minus 1, then two sequences are aligned just by chance, therefore no homology. Since the p-value in the present case is approximately equal to 10 raised to the power minus 6, and this falls between 10 raised to the power minus 5 to 10 raised to the power minus 5 range, therefore the two sequences involved in present alignment are having clear homology. Choose lower PAM number matrix with high gap open and high gap extent penalties for aligning closely related sequences. For aligning distantly related sequences, choose higher PAM numbers matrix with low gap open and low gap extent penalty. When the divergence distance is not known or the sequences are having intermediate divergences, choose PAM 160 with 10 for gap open and 1 for gap extent penalties. Alternatively, try again all the three options to arrive at 3 alignments to select the best one. Finally, click Submit button. The HPR protein sequences from E. coli HPR and E. faecalis HPR in FASTA format and their length 90 and 88 shown respectively here in this slide. Let us analyze these two sequences using dot plot with the Dotter program for sequence alignment. To visualize the extent of sequence identity between these two sequences, let us use Dotter software first. Download Dotter if you have not already downloaded Dotter. To download, see dot plus analysis for visualizing repeats in protein sequences in the module on protein sequence analysis. To use Dotter, run Dotter on MS DOS prompt by typing Dotter followed by space E. coli HPR.fa, that is the name of the first file containing sequence, followed by space E. faecalis HPR.fa, the name of the second file containing HPR from E. faecalis, and press enter. Dotter main window will appear. Use context menu which will appear with a right mouse click in MS Windows operating system. Display window size tool and gray ramp tool. Set the window size to 30 and adjust the gray ramp tool values between 29 and 36. Try other values also to make the graph without background noise. This main window shows that the two sequences are having identities in the N-terminal and C-terminal regions with non-identities in the middle portion of the sequence. Display dot plot alignment window using context menu. This shows that the two sequences are having identities, and these identities are highlighted by cyan color in the N and C terminals with the non-identical region in the middle. The histidine at position 60 involved in the function of HPR of E. coli is aligned with the histidine at position 15 of HPR of E. faecalis. However, the serine at position 48 involved in the function of HPR of E. coli is aligned with isoleucine at position 47, and serine at position 48 is not aligned with serine 46 at position 46 in HPR of E. faecalis. Therefore, user need to align these two sequences, that is the HPR proteins, using a specialized sequence alignment program. Embossed structure alignment between HPR of E. coli and HPR of E. faecalis using BLOSSOM 62 with opening gap penalty 10 and extend gap penalty 1 was constructed. This alignment reported 23.3% identities for the overlap alignment length of 90 positions. The histidine and serine residues, which are involved in the functioning of HPR in different pathways, are aligned in the same columns. The 23.3% identities for the overlapping alignment length of 90 positions fall in yellow color twilight zone. Therefore, it is not safe to infer that two sequences are homologous from this XY graph. Consequently, we need to calculate p-value using BLOSSOM 62 and opening gap penalty 10 with extend gap penalty 1 for evaluating the alignment. P-value for this alignment was found to be 8.1 into 10 raised to the power minus 11. This p-value fall within the range of 10 raised to the power minus 50 to 10 raised to the power minus 5. Therefore, these two sequences are having clear homology from p-value table. Consequently, statistically, it is safe to draw an inference that the two sequences are homologous sequences. The alignment reported 23.3% identity, which is nearly equal to 25% sequence identity. This shows that approximately 25% sequence identity, and for this level of identity, the two sequences appears to be distantly related. Therefore, PAM 200 with low open gap penalty of 1 and low extend gap penalty may be tried for sequence alignment to infer structure alignment. The alignment between HPR from E. coli and HPR from E. faecalis is using embossed structure. Using PAM 200 with opening gap 1 and extending gap 1, calculated 31.0% identities for the 113 alignment length with a score of 152. This alignment length falls in green colored safe zone. Therefore, it is safe to infer that the two sequences are homologous. Consequently, the knowledge about HPR from E. coli can be applied in understanding the structure and function of HPR from E. faecalis. And serine 46 in E. faecalis. Therefore, the alignment appears to be functional alignment and can be used for function assignment. However, the increase in overlap length of global alignment from 91 residues to 113 residues may misguide for structure alignment. With increase in overlap length of global alignment from 91 to 113 residues, it is necessary to know if the alignment itself indicates a biochemically significant alignment for assigning structure also. Therefore, let us use Smith-Waterman algorithm of dynamic programming between full-length sequences of two unequal length proteins. The E-value shows that the two sequences are having clear homology. Consequently, statistically, it is safe to draw an inference that the two sequences are homologous with 87 amino acids overlap. Subsequently, local alignment of S-search was compared with global alignment using embossed structure with parameters blossom 62, open gap 10, extend gap 1. The sequences in local alignment are labeled SW_EC for HPR sequence from E. coli and SW_EF for HPR sequence from E. faecalis. The global alignment is labeled with NW_EC for HPR sequence from E. coli and NW_EF for HPR sequence from E. faecalis. The residues histidine and serine are important for its function in two bacteria, and these are aligned in both the alignments. Therefore, these alignments are meaningful for inferring functional homology between two proteins, which indicates that both the proteins might have similar structure also. Solution structures of the enzyme 2A HPR complex of E. coli phosphotransferase system has been determined experimentally and is shown on the left side. Three-dimensional structure of HPR protein of Enterococcus faecalis has also been determined experimentally and is shown on the right side of this slide. This structure is similar to HPR of E. coli shown in the complex. However, the structure of enzyme 2A HPR complex of phosphotransferase system of Enterococcus faecalis has not been determined experimentally. Since the two HPR proteins in both the species has been found to be homologous, therefore structural knowledge which has been determined experimentally for enzyme 2A HPR complex of E. coli can be used for modeling uptake and phosphorylation of specific sugars by enzyme 2A HPR complex of Enterococcus faecalis. Dear students, a histidine phosphorylated HPR protein is used by E. coli for uptake of carbohydrate through PEP-dependent phosphotransferase system. The histidine residue of HPR is phosphorylated using PEP. In addition, this HPR can also be phosphorylated at another residue, a serine, but using ATP. Serine phosphorylated HPR acts as an intermediate in the signaling cascade, which regulates transcription of genes involved in carbohydrate metabolism. Both functions involve a phosphorylation reaction but at different residues on the HPR and are involved in different pathways. Histidine phosphorylated HPR acts at cell membrane for uptake of sugars, and serine phosphorylated HPR acts on DNA for regulation of expression of genes involved in sugar metabolism. In addition, serine phosphorylated HPR does not allow phosphorylation at histidine, thereby blocking the sugar uptake. The HPR is also present in Enterococcus faecalis genome. Let us use the sequence of HPR from Enterococcus faecalis to align HPR from E. coli to see if these proteins are homologous. In case they are homologous, then all the structural and functional knowledge about HPR in E. coli can be applied for modeling sugar uptake and transcription regulation process in Enterococcus faecalis. Therefore, let us use these two HPR proteins, that is one from E. coli K12 and other from E. faecalis V583. Download these protein sequences and save in separate files named E. coli HPR.fa and E. faecalis HPR.fa respectively. Dear students, the major goal of sequence alignment is to find sequence similarities so as to provide a basis for function conservation between two sequences. The function can be conserved only if the structure is also conserved. Homology refers to similarity due to descent from a common ancestor and indicates that known knowledge about the structure and function of one sequence can be applied to infer structure and function of other sequence. In the practical example, we used HPR proteins from E. coli and E. faecalis and found that the alignments between these two proteins, that is HPR proteins from two different species, were meaningful for inferring homology between these two proteins. Consequently, the structural and functional knowledge which has been derived experimentally for HPR in E. coli can be applied for modeling biological processes, that is uptake of sugars and regulation of gene expression involving the HPR in E. faecalis. Consequently, the sequence alignment provides the basis for identifying similar sequences, consequently similar structures, and finally similar function. I thank you for visiting EPG Pathshala.